It's my pleasure to introduce Rene, and I worked in his lab for 4.5 years, more or less, almost by accident. But this is a story for another day. And I've learned a lot in his lab because when I started there, I didn't know anything about transposons or small RNA processing, or very little about that. But let's back to the beginning. So Rene is currently director at the IMB, a fairly new institute in Germany, in Mainz, but a very successful one, I think, considering its age. Rene did his PhD in the Netherlands, and he followed by a postdoc in Cold Spring, Harvard, and then back to the Netherlands, to Utrecht, where then he became a professor before moving to Germany. And all of his research, if I can summarize it, has been on small RNA processing, going back in the day, for people who still remember microRNAs, the microprocessor, I believe it's called the microprocessor component that he studied, and then going all the way to piRNAs. And piRNAs are fascinating because they are small RNAs, non-coding RNAs, that stop transposons from basically invading our genomes. And this is super cool because without that, we basically wouldn't exist, because it would just destroy our genome integrity, and we wouldn't be able, especially in the germ cells, there would be no reproduction at all. And this is one of the things I've learned in his lab. And hopefully we'll talk more about the germ granules and the small RNAs in a much better way than I can do, because I didn't learn enough back there. And he will also hopefully show some beautiful C. elegans pictures, which I admire a lot. So with that, because I can't do justice to the work that you've done, Rene, please, the floor is yours.
Yeah. Thanks, Antonio, for the introduction. And thanks for the opportunity to talk about our work here on this platform. Of course, this platform is focused on all flavors and types and properties of condensates, and this is also a field of interest that has our strong attention, because the small RNAs that Antonio mentioned are typically active and are also being made within condensates. And sort of the bigger picture of how we sort of see that interaction between these two fields now is that we would really love to use the knowledge of the small RNA pathways that we are studying, not only to just understand how the small RNA pathways are working, but also to understand how these condensates, how these germ granules are really working, really in a biological setting that has a lot of relevance to fertility, to oogenesis, to spermatogenesis, and hence with a lot of biological relevance there. And I think the system that I will talk about offers a lot of opportunities for that. For instance, what you see here, so this is one of our two model systems, C. elegans, at least a fraction of it. And what you see here, this structure right here that goes around the corner, is the germline of C. elegans. And you see in that germline, you see these green blobs or granules, and these are the P granules. Of course, I would assume that they are very well known by all of you that are attending these. This is, I would say, one of the best studied structures in that in this field, I would say. Now, these are P granules, and actually, I will talk about a novel type of germ granule that we identified not too long ago, which is actually not visible here, because it does not contain this protein that we've labeled here, and it's present in this little structure right here, which are the sperm cells. This is actually this organ that you see here is a spermatheca that stores the sperm, and whenever an oocyte is, so this is an oocyte, is pressed through the spermatheca, it's fertilized by one of the sperm, and of course oogenesis starts. And I'll talk about a dedicated condensate in these sperm cells, about the biological function. That will be the first half of the talk, and then in the second half, and that is largely published, the second half I would go into efforts of us to study these novel granules at a more biochemical level. But this is still early days, and I hope maybe to also get some good suggestions from your side. Why does this keep happening? Okay, like this. Okay, so just briefly, these small RNA pathways, they are characterized by proteins that you see here revolving. This is a 3D structure from the lab of Dinchal Patel. This is an argonaute protein with a small RNA bound. So the red stuff is the small RNA. And really, it's this combination of the argonaute protein with a small RNA that makes the biological relevance. The small RNA alone would be highly unstable. The argonaute without a small RNA, as far as we know now, has hardly any function. But together, they form a very potent gene regulatory module, by which the small RNA recognizes in a sequence-specific manner a target, and the argonaute protein then triggers an effect. And such an effect could be translation, typically the inhibition of translation, but also RNA stability. And it could even attract factors to chromatin to modify histone tails, for instance. I'm sorry, I don't know why this happens. So small RNA pathways are highly conserved. They're found in all plates of life, or all domains of life. And whenever they are there, they're highly relevant for either viability or typically also the fertility of the animal. So like in animals, there is this clade of Pee-wee proteins that is highly germ cell specific. Antonio briefly mentioned that. And also in C. elegans has a Pee-wee protein, but C. elegans also has its own sort of evolved set of argonaute proteins that are mostly, as far as we know now, either specifically expressed in the germline or at least highly enriched in the germline. And it's this class of proteins that I'll be talking about. And it's this class of proteins that we are trying to use to really get the different functions of the RNAi pathways, but also of the condensates, because of like there's here, it's indicated there's like 13 different ones. And they show very interesting differences in their localization, and we start to understand a little bit about their functions, their specific functions as well. So I want to talk a little bit about, I want to talk today about inheritance of silencing, so of these small RNAs, and in particular through the male, and just to show, inheritance of silencing in C. elegans is heavily studied. It's been proposed to play a role in many different processes, and this is, for instance, a paper from the Murphy lab that has to do with inheritance of experiences of the parents encountering pathogens and then transmitting that information to offspring in the form of small RNAs. And this is a paper from the lab of Odette Shafi showing behavioral effects that can be sometimes transmitted across generations. But the problem with these and many studies is that the relevance, sort of there are effects, but the molecular mechanisms that are behind it are really not well understood. And I think we really need to get to those molecular details before we can really be certain of how to interpret these studies. Now, this is a paper from the group of Andy Fire, one of the discoverers of RNAi, and they already noted very early on that RNAi can be inherited, and it can actually be very effectively inherited through the male gametic line, as it's written here. And this is remarkable because, I mean, the oocyte is of course big, has a lot of cytoplasm, and a lot of material is inherited. And of course, we all know about maternal effects, proteins and RNAs that come via the oocyte. And the sperm typically doesn't have that because a lot of the cytoplasm is kicked out of the spermatocyte during development. So you end up with these very small spermatids, and a lot of the cytoplasm is kicked out into this residual body. And this is not specific for C. elegans. This happens also in mammals and I guess virtually all systems that make sperm. And one of the functions is assumed is sort of to make the sperm as small as possible. But this, so there's a lot of the material is being kicked out of the sperm. And so Jan, PhD student, or now a postdoc in the lab, asked the question, is there a dedicated mechanism maybe that goes against this process of kicking stuff out of this developing spermatid so that you can actually inherit small RNAs via the male. And this was initiated by studies that he did on one of these, one of these big clade of Argonaute proteins in C. elegans, the WAGO proteins. He studied WAGO3. You see a very similar picture as I showed you in the beginning of the P granules. These are indeed P granules. These are all nicely overlapping with this P granule marker PEGL1. But then you see the difference here. In this case, you see signal here in this region where the sperm is made and stored, whereas the PEGL1 marker is not expressed there. And so we wondered, so how can WAGO3 protein be, how is it specifically retained in that sperm? And so when we looked at the process of spermatogenesis, so here you see a gonad of C. elegans that is actually doing spermatogenesis. I will not bore you with all the sort of the biological details there, but here spermatogenesis is going on, and this animal is expressing both PEGL1, which marks the P granules, as well as WAGO3. And you see here in the beginning, there is nice co-localization between the two. And then here you see the PEGL1 signal starts to separate away from the WAGO3 signal. And you get these distinct foci of WAGO3 that end up in sperm. And I would show you this diagram, this image that I showed you before. So this is the development of a primary spermatocyte to mature sperm. And here you have this stage where you have this residual body, where all this material is being kicked out, but the WAGO3 granules clearly are retained within the developing sperm. And so WAGO3 is inherited, is loaded into sperm. So does it have an effect on inheritance? The short answer is yes, it affects inheritance of gene silencing via the sperm very effectively. And this is, I just want to show you one assay that we did that looks at this in an endogenous setting. So we are not triggering silencing here by double-stranded RNA, but this is all endogenous. And so what you see here, this is a cross between two wild-type animals, a male and a female, or a hermaphrodite. And of course, the offspring is again wild type and is again fertile. The thing is, we can take out many of the small RNA pathways, and there are many defects, but typically the animals are still fertile. So you hear the male is defective in these two pathways, the female also, the offspring of course also. There are problems, but it's not so bad. The problem really starts when the parents do not have these small RNA pathways, but the offspring has. So in this case, the offspring needs to initiate the small RNA pathway. So it needs to initiate the pool of small RNAs that it's using for silencing without any information, if there is any, from the parents. And apparently there is a lot of information from the parents, because if the parents don't have this, the offspring don't know what to do. And they make small RNAs against all sorts of genes that actually are needed by the germ cells. And so the animals develop into sterile animals. And this is a phenotype that we call mutator-induced sterility. And we use this to look at inheritance of endogenously required small RNAs. And I'll just show you these sort of diagrams every now and then, not too many. So this shows this sterility effect. This blue bar here, 70% of sterility. And here you just show that WAGO3 is specifically required in the male to prevent this sterility. So here in the WAGO3 mutant male, a WAGO3 mutant father sires offspring in this setting that is completely sterile. Whereas if it comes from, if the mother is WAGO3 mutant, it doesn't really care. So WAGO3 is really, I mean, if this went above the genetics, that's it's a complicated setting. It shows that WAGO3 is really important for the inheritance of small RNAs via the sperm. And so the question we started to ask is, how is WAGO3 maintained in this mature sperm? And we have seen these granules, and this study was propelled forward by a finding that Jan did when he did an IP mass spectrometry experiment where we identified this P1 protein, standing for paternal epigenetic inheritance. And P1 is a protein schematically like this. It has a BTB and back domain at its N terminus, and for the rest, it appears to be rather disordered. And P1 is highly specifically expressed just at the later stages of spermatogenesis. So here again, you see a spermatogenesis arm of C. elegans going from left to right. So here you have early stages, and here you have late stages. And P1 here is expressed only in these late stages. And we haven't seen any expression anywhere else. And it recruits WAGO3 into these granules. So WAGO3 and P1 basically always co-localize. Whenever there's the P1 is expressed, WAGO3 is also expressed in the same granule. But there are also, I told you, there's this large family of organelle proteins. There's about 13, and a number of them are also expressed during spermatogenesis, such as WAGO1 and ALG3. They are quite like WAGO3. WAGO1 is actually highly homologous to WAGO3, but these two proteins do not go into these granules. So they go into these residual bodies that you see here. So here indicated this is the residual body. This would be the sperm. So these granules are not only interesting because they are specifically going into sperm and recruit WAGO3, but at the same time, they're also highly selective. And this is, for longer term studies, that's what I would get to at the end. We want to understand how the selectivity is achieved in these condensates. Okay, so this is showing at another stage. So this is this budding spermatid stage where you see here this residual body and here the spermatids. And just to show you a bit clearly, these granules are really specifically in sperm and not in these bodies. And if you take away the P1 protein, so WAGO3 is still there, but P1 is not there. This is the wild type that I've just shown you. In the mutant, WAGO3 is basically completely lost into the residual body. So you see that WAGO3 is not by default maintained there. It needs the P1 protein. Presumably it needs these granules. And the P1 protein, just like WAGO3, is required for inheritance of small RNA information, also in genetic assays. That's the one I just explained to you. So I want to get a bit more mechanistic here in terms of how these granules are then kept within the sperm. Why don't they go into the residual bodies? And for that, I need to briefly explain to you, introduce to you what is happening during spermatid formation. So during development, during these meiotic divisions, this organelle forms. So this is called fibrous body membranous organelle. It's an organelle that derives from the Golgi. It has some membranes and it has some fibrous proteins that are getting in there. And this organelle at this stage looks like this octopus. And it has already been described years ago that indeed these organelles are transported selectively into the mature sperm and do not end up in this residual body. And later in the spermatids, they have specific functions. Forget the right part of this graph. So the sorting of these FBMOs, so this process right here, that they are not here, but they are all in the sperm, depends on myosin 6, which is in C. elegans called Sp15. And so we looked in Sp15 mutants at how the P granules are behaving, and sure enough, we saw that in the wild type, they are nicely out of the residual body. In these myosin mutants, they basically are all over the place, as if they are not sorted properly. So we started to think, maybe they are attached to these FBMOs in some way. And one way to look at this, we collaborated with the microscopy facility at the EMBL, doing CLEM. And so here you see electron micrographs of early stages of spermatocytes and later stages all the way to single spermatids. Sort of labeled the different organelles that you can recognize here. So here you may recognize this FBMO organelle, sort of the same shape as here. And if we overlay now the P1 granules, they basically always are right on top or right next to these FBMOs. And so our thinking was and still is that these condensates have formed contacts with these FBMOs. And because the FBMOs are being transported into the mature sperm, the P1 granules are also transported into mature sperm. So how are these condensates then, how may they be attached to these FBMOs? And that is, I'll show you some data that is, we believe, through this protein that we call P2. So P2 was identified in an IP mass spec, in an IP mass spec experiment. P1 was highly enriched and P2 was one of the top hits. P2 has basically an identical expression pattern as P1, only during spermatogenesis, always in these granules, always in sperm and not in the residual bodies. And if you now delete P2, you still have granules. There are fewer, and we haven't fully characterized, their properties yet. But the striking thing that we observed is that they look like this myosin mutants. They're basically all over the place. They don't appear to get sorted. And so we interpret this as that P2 may be forming a link between the P granule and these FBMOs. And sure enough, P2 has this same phenotype as WAGO3 and WAGO1. So genetic incapability of paternal inheritance. So this is how we think about these sort of like a schematic of these P granules. There's these FBMO membranes. We have the granules that are formed by P1 and where also P2 is present. They can selectively recruit WAGO3, but they would not take up related argonaute proteins such as WAGO1. And then, of course, the question is, how are these then connected to these membranes? We don't have a full answer, but we have an indication as to what may be happening. And for that, I want to show you the phenotype of a mutant, which is called SPE10. SPE10 is a palmitoyl transferase, so it transfers fatty acids onto proteins. And it's specifically found on these FBMO membranes in sperm. And in SPE10 mutants, these P granules, they start to form, but then they aggregate together in much larger structures that are no longer nice and round, and they end up into residual bodies. So we think they are not attached anymore to the FBMO. And indeed, we have evidence that P2, so you see here Western blot of P2, this would be the unmodified size of P2. And this is a modified version that we always detect of P2. And this is basically, this higher band totally depends on the presence of this palmitoyl transferase. So within the SPE10 mutant, we don't have this modified form, we have only the unmodified form. And so we think that P2 may be palmitoylated by this enzyme SPE10, and that these fatty acid tails may provide interaction modes with the membrane. And this is something that, of course, this is no direct proof. Personally, I would think, if such a link would be strong enough to sort of act in such a way, but at least the genetic data seems to point in that direction. So this is for the published part. What is happening is that in naive germ cells, you have a lot of argonaute proteins. One of them is WAGO3. And they are typically found in these P granules. I haven't talked about these. I mean, there are also, people have also described other types of condensates that are typically very close to P granules. But in that respect, I would like to keep it simple. So they are in the P granules. During spermatogenesis, WAGO3 is leaving the P granules, and the P granules are also disassembling. We don't know what is cause or consequence. It may simply be a disassembly of the P granules that makes WAGO3 leave. And at the same time, these pay granules start to form, and they form on the membranes of these FBMOs. And they take up WAGO3, but not other proteins such as WAGO1. And then this P15 protein, myosin-mediated transport, makes sure that this complex is retained in sperm and is not discarded in the residual body. Then, of course, and then we get into a topic that we haven't studied yet, but is also, our prime interest we are starting to look into that. So then you have a single spermatid that develops into a spermatozoon. Of course, that would develop that would fertilize an oocyte, and to be active, then of course the WAGO3 protein would need to be released from these pay granules. And how this works, we don't know. Possibly simple dilution, because of the big oocyte and the small sperm, that could be a fact. But we also are not excluding post-translational modifications that to play a role in this process. But and then the released WAGO3 can act in the zygote to do its thing. Okay, so the questions that we are now trying to address, and we are doing that not alone. We are also collaborating heavily on this project together with Lucas Stelzel here in Mainz. Is how do they form the pay granules? What is the role of this BTP back domain and the IDR? What do the different domains do? And how is the specificity achieved towards WAGO3? And then in a later stage, but I will not touch upon that, how do they then release WAGO3 in the end in the oocyte? So I've shown you this little diagram of the PAY1 protein. It's a pretty simple protein. BTP back are two folded domains and a large and an IDR at the C terminus and a little IDR at the N terminus. And what we've done before, I didn't go into details there, we have genetically deleted different domains of the PAY1 protein and looked at the effects in vivo. And what we saw is that, so this would be wild type, the left part here. The middle part here is when we delete the BTP and back domain, but the IDR is still there. And what we observe is that the pay granules are very less, they're much less intense. They're typically very small, but they still contain WAGO3. So WAGO3 is still there. So we think that the BTP back domains are driving the assembly of the granule, but are not involved in the WAGO3 interaction. And the other way around, if you delete the IDR, the pay granules still form. You see here these like the purple granules, but it loses the propensity to recruit WAGO3. So the BTP back domain, we think, is a scaffold to sort of make to polymerize maybe. Whereas the IDR is not driving here the condensate, but seems at least not on its own, but it's more recruiting the WAGO3 protein. And so, but this is still in vivo, and we want to make the step to in vitro studies, to understand these condensates in a bit more detail, to really be able to explain the observations here, because of course here there's a lot of factors that are still affecting these granules, and every mutant that you make can have indirect effects. So in principle, a bit hard to interpret. So Diego, a postdoc in the lab, has been picking up this project, and he's been purifying PAY1 and PAY2 proteins, full length in first instance. He gets relatively nice proteins and tries to do phase separation assays. And what we see is that if he only takes the PAY1 protein, we get very little units formed. PAY2 doesn't do anything. It seems to be a very soluble protein. But when we mix the two, we get larger condensates formed. And if I just blow it up a bit for you, so we get these larger units that are not nicely round, right? I mean, you can clearly see that. I guess, in the first guess that I would say, they're maybe a bit more gel-like or so, but we don't have material properties on this as yet. And you may already see some sort of substructures here. And this is something that Diego has seen. If he labels P1 and P2 with different dyes, we see that P2 typically forms the most of the granule, and the P1 proteins seems to form foci within these larger units. Now, this is something that we have not, well, we haven't gone back yet in vivo to see whether substructures exist also in the P granules in C. elegans. That will be difficult also, because sort of the size bar here is 10 microns, and 10 microns is basically the size of the complete spermatid. So these structures here are clearly much larger than what we have in vivo. But it could still be an interesting, I think it's an interesting lead for us to sort of see whether this sort of formation of substructures, of subdomains, may apply to P granules in vivo as well. And of course, what the relevance of that then is. Now to the BTB back domains. BTB and back domains have been described, and there's this very nice paper from the lab of Tanya Mittag. She showed in yeast, in this protein SPOP, that the BTB and back domains form a polymer where the BTB domains dimerize and the back domains can dimerize. So you can get this polymeric chains, and you can different chains can form together to form larger condensates. So does this also apply to our proteins? And we are studying this now. This is what Diego is currently doing. He's doing a lot of different studies using either purified proteins, but also yeast to hybrids to see which BTB domain could bind to which other domain, and vice versa for the back domain. And so far, just as a summary, without going into all the data points there, P1 seems to be able to polymerize, as I showed you for this case here. So the BTB domains can dimerize, as well as the back domains can dimerize. For P2, it's different. P2 only seems to be able to dimerize via its BTB domain, so you get only very short units. And I guess this would be consistent with what we see in the phase separation assays, where P1 on its own can do something, but P2 seems to be very soluble. And then testing all the combinations, it seems that P2 can sort of fit into this P1 polymer chain very easily in many different combinations. Why this then drives the condensates to the extent that we see, we don't know. And of course, this is something that Diego is now further following up on. And one thing that we are trying to do there, also to sort of go back in vivo and test, is this relevant at all for what we see in vivo? Now, using AlphaFold, we seem to be getting very nice predictions of the BTB interactions, and can predict how a P2 interaction would look like, and a P1-P2 interaction would look like. And we are making mutations in the residues that appear to be involved. At least in yeast to hybrids, these residues are involved. But we have not yet been able to test such mutants in vitro phase separation assays or in C. elegans. Then, as a final part, so this BTB back domain story, I think, really forms the backbone of the P granules. It's a polymerization domain, and P1 and P2 clearly co-evolve to sort of be able to sort of form mixed chain polymers in that sense. Then I want to briefly touch upon the specificity and what we have learned about how WAGO3 could be recruited. I told you that the P1 IDR is required, right? So if you don't have the IDR on P1, you lose the WAGO3 into the residual body, and WAGO3 really does not accumulate in the P granules. Now, what about the side of WAGO3, right? So WAGO3 is, for the larger part, a folded protein, indicated by this very simplistic ellipse. And it has an N-terminal intrinsically disordered region, which is very proline rich. So one thing that we did is we took out the IDR, but at least in vivo, this creates a dominant sterile phenotype. And dominant sterile is not something that you can really work with because already the heterozygotes are dead or are sterile, so we can't really analyze it. And so we are making inducible systems. A PhD student in the group is doing that, and we can indeed now induce the expression of IDR-less WAGO3. We induce that phenotype, and we are going to study how WAGO3 then behaves. But we are not far enough there to sort of draw any conclusions. What evidence that why we think that the IDR of WAGO3 is involved in recruitment to the pay granules comes from a collaboration with Lucas Stelzel and his PhD student Kumar Gaurav. They're doing dynamic simulations, and this is one where you see individual IDRs change of pay1. The simulations in the coarse grain simulation here, they do form condensate. So this is something that we may have expected, of course, but it was good to see. And then we proceeded to test, does the WAGO3 IDR like to be in these condensates, yes or no? And how about the WAGO1 IDR? And the answer there was that clearly the WAGO3 IDR likes to be in these condensates, whereas the WAGO1 IDR, it doesn't, let's say, it doesn't hate it, but it doesn't like it as much as the WAGO3 IDR. But this is still a very vague term. This is very much work in process, but we think we may be onto something here that there is sequence characteristics within the IDR that make it go into the pay granules for WAGO3, but not for WAGO1. And you see this also in a simulation here, where you see in green the pay1 IDR forming condensates, and in red you see the IDR of WAGO3, and you see it nicely joins these condensates. So we think that the WAGO3 IDR has an important role in going to these granules, and actually we think that the dominant sterility that we see is related to other condensates, where the IDR may also regulate, where WAGO3 goes, because what we think is that the WAGO3 without the IDR is actually loaded with the wrong type of small RNAs. So it may be present in the wrong condensates and thereby getting the wrong small RNAs. So also for that, we really need to understand how, the IDRs of the WAGO proteins are determining which condensates they are joining and which ones not. And a final indication in this direction is a final set of experiments that I'd like to show you. We have seen that the IDRs of these WAGO proteins are processed. So this paper here, we provided a little help to the lab of Helge Gooshans, who published a nice paper on this protein called DPF3, which is a protease. It has an activity that cleaves like 2 amino acids from the N-terminus, provided that the second one is a proline or an alanine. And DPF3 has very strong effects on WAGO3 and also on WAGO1. And they could show that the IDR of WAGO3 and WAGO1 is actually processed by DPF3. And I want to briefly introduce you a second enzyme called APP1, which is very similar to DPF3, but its activity, its substrate specificity and its activity are slightly different as depicted here. And this enzyme we identified in an inheritance paper where we were looking at factors that were required for inheritance of RNAi. And so both of these proteases, both DPF3 and APP1, they localize to pay granules. So here you see the pay granules labeled by pay1, here you see DPF3, and here you see APP1. Clearly, APP1 is not fully recruited to these pay granules, but there's clear signal there. And genetically, I'm not going to show you the data. Genetically, both APP1 and DPF3 are required for inheritance of silencing via the male. So then we start to ask the question, so if the processing of the N-terminal tail is important, we should start to look at WAGO3 and other organelles, not with the GFP, not at the full N-terminus, because it would block processing, but at more internal sites. And so this is what Ida, a PhD student in the group, is doing. Here we have a WAGO3 with an internally tagged GFP. And you see it localizes to the P granules. And then in absence of these two proteases, we completely lose the localization of WAGO3 to these P granules. So processing of the IDR, shortening of the IDR, greatly stimulates the participation of WAGO3 in these granules. And we currently try to understand, why, how such trimming of the N-terminus could help explain this phenotype, and how in vivo this type of processing may be used for specificity causes to determine what WAGO protein will go. So finally, this is my last slide. How we think that the P granules are working is we have a backbone of BTB back domain interactions of these two proteins called P1 and P2. The IDR of P1 we think forms a brush that allows interaction then with the IDR of WAGO3, but only if the WAGO3 IDR is properly processed and trimmed. So here you see a shorter version. A longer version does not like to interact with this IDR brush. At the same time, P2 we think is palmitoyl-related, and that provides an anchor to these FBMOs. And I didn't really show you these, but we also have indications that this tethering to a membrane actually brings post-translational modifications also to the P1 protein. We just don't know what these modifications are at the moment. And so by trying to understand this one germ granule in much greater detail, and I've shown you what we are trying to do, we hope to use the small RNA pathways to really understand how condensates can be used to segregate out very specifically specific molecules and secure them in some place of the cell. And I haven't talked about this. I mean, this type of protein is conserved. You find it also in human genome. Not specifically, not very directly, but BTB back domains followed by an IDR are proteins that you clearly see, and so we think that this type of process may drive subcellular organization also in other systems than C. elegans spermatogenesis. And so with that, I've come to the end, and I would like to thank the people I have mentioned, Jan Schreier, who has done the work. I mentioned Diego. Diego didn't join us on this particular retreat. He's the postdoc who's doing the in vitro work now. And Ida and Lisa are following up on the IDR biochemistry and genetics of WAGO3. And I'd like to highlight here our collaboration with Lukas Stelzel and Kumar Gaurav, which is leading to the identification of really interesting motifs that may or may not explain the observations that we are seeing in vivo. And with that, I'd like to stop and take any questions or suggestions on how we can better understand our P granules. Thank you very much.
Thank you.
Thank you. So there was a question in chat, Rene, from Shruti. So Shruti, would you mind asking the question?
Yeah.
Yeah.
Thanks for the talk. Really nice talk. I was just wondering, since as much as I understood, these are RNA binding protein, and I was wondering if you have looked up about, tried to change the availability of the RNA, or try to silence the, remove the RNA that these proteins bind to, and then try to see where they localize to, and if their ability to form condensate is affected by presence or absence of these RNAs.
Yeah, sure. So WAGO3, sorry, I have a strong echo here. WAGO3 is indeed an RNA binding protein, right? It binds these small RNAs. Without the small RNAs, WAGO3 is mostly very unstable. So it doesn't accumulate at all to any appreciable level. And so it also doesn't join the pay granule. That said, the pay granules themselves, as judged by pay1 or pay2, they don't rely on WAGO3. So you can take out WAGO3, but these foci that are formed by pay1 and pay2, they are unaffected. So in that sense, the small RNAs don't contribute to the condensate formation, which I guess is not very surprising. Whether there are larger RNAs in there, at the moment we just don't know.
If they're unstable without small RNAs, then how can we say that they do not contribute in condensate formation? Does it not require to be like stable in that sense to localize to the condensates? Like, so you mean that the WAGO3, even in absence of small RNA, they do participate in the condensate?
No, because we can't see the protein. At least so I don't know. I mean, the few proteins that are there, we just can't see them because they're just too few.
Okay.
So it's that said, so there is an RNA binding. So the IP mass spec that we did on pay1 did identify a protein with an RBM. It was very, very high up in the list. I think it was even most high up. And so far, we just don't know the function of that protein and whether it indeed binds RNA and if that contributes to pay granules at all. So,
okay, thanks.
Yeah.
Thanks.
I will also follow up with a question since we don't have any more in chat. But I'm curious, you mentioned that there was a difference between the WAGO1 and the WAGO3 terminal, the IDR. And could you comment a little bit further? Because I believe the WAGO3 was rich in prolines.
They both are. They both have this very proline-rich sequence. The WAGO, the net charge, so if you look at the overall charge not the distribution, the overall charge of the WAGO3 IDR is a bit different from the WAGO1 IDR. And that difference gets stronger when you process the end-term, like that IDR, when you make it shorter from the end-terminus. So it could be that charge difference plays some, but we have not looked at. We have not been able to identify, like stickers or any type of that sort of thing, and sort of how this interaction would take place. This is, I'm afraid, a bit too early in those stages. But so there are differences, but it's nothing spectacular.
I guess one experiment would be the reverse. So tag the tail of WAGO1 in WAGO3 and see what happens. Yeah.
I mean, that's something that that we have considered, of course. Hybrid protein. I'm not the biggest fan of making such hybrids, but I think in this case it may actually be a good thing. We just have not gotten to do that. I think it's
Jill, any more questions on your side?
I don't know. Anybody at the kitchen table? Did I not see anybody? I mean, I can ask a question because I'm a big fan of BTB and back domains, as I used to be in Tanya's lab. So
I didn't know.
we also have I'll hold mine. I see there's an Ashish with a hand up. Do you want to unmute yourself?
Yeah. So one thing I wanted to ask was, have you looked at the mobility of these proteins, maybe through FRAP recovery or something? Maybe that would show like how these. Hello.
Yeah. So we have. You still hear me?
Yeah. Can hear you.
Oh, yes, we have done that. I mean, I haven't shown that. So we have looked at FRAP of WAGO3, and we have done that in the in the pay granules as well as in the P granules, right? So we did it in the body of the germline as well as in the sperm. And the recovery of, I don't recall the the exact recovery time, but at least the recovery in the P granules was significantly faster than in the P granules, which would be consistent with these P granules, which seem to be sort of doing transport of some sort, having really a different substance maybe than the P granules. One being maybe more liquid, and the other maybe being more gel-like. I'm a bit cautious there because we just didn't do any of these sort of assays. I think that people advocate that you should do, and I think that are fair to do, to really make statements on what these materials could look like. But Wago 3 is definitely less mobile in the P granule compared to a P granule.
Right. Oh, and maybe like the PTM subspecies could drive that kind of difference in mobility.
Sorry, again, you were breaking up a bit.
Okay. Yeah, I was just saying that the PTM species, subspecies of these proteins could drive that kind of heterogeneity.
Yeah.
Okay.
Yeah.
Then another thing I wanted to ask is what about the molecular grammar, sort of like the what kind of patches are increasing the preference of Wago 3, and what are excluding that? For example, you have acidic patch or just polar residue patch that sort of create that environment.
Yeah, no, so we have played a little bit, but only a little bit, with the IDR of P1. We added a glycine and serine rich region of Perl 1 and stuck it onto P1. So and that made it, that made Wago 3 move quicker. So the frap times went down. But again, I don't really know how to really interpret that, other than when you change the IDR, you change its properties, which I think is not very surprising. So we have not been able to really make strong statements on like some sort of combinations or certain spacings of amino acids. And within Wago 3, really the only thing, there's lots of serenines and threonines. And there's ample opportunity for phosphorylation there. We have identified actually a kinase also in this whole process that seems to be acting there. So I think phosphorylation would definitely play a role. And as Antonio said, there's tons of prolines. It's a very proline-rich IDR. That is really the first thing that you notice. When you open that sequence in Wormbase, you're like, oh wow, this is. So that is really the one thing that stands out. We haven't been able to find any other particular enrichments or depletions.
Okay, yeah, these are actually very interesting observations that you told me about. Okay, thank you.
Loving the discussion, guys. I guess I'll ask mine now. So I was really intrigued by the BTB back interface and the PEI1, PEI1, 2, I don't know. So I was interested that the back domain, was it the BTB or the back that didn't dimerize in PEI2?
In PEI2, the back domain doesn't dimerize, no.
Did you guys look bioinformatically and see why that is?
So we try to do that with now with AlphaFold, yeah. So we don't at the moment. We just don't know. We just know that the BTB and the back domains of P1, they seem to be pretty promiscuous. They can do it with with other P1s or with P2 that can mix and match.
Are they structurally similar? And that's
they're very similar. If you look at the predictions, they're very similar, but clearly not identical, right? The sequence is not identical, and also if you look at the folds, at the predicted folds, these are not identical. And so what I would like to do, and what I think would also, what I hope that we can do is that if we can make nice monodisperse dimers by mutating some of these interfaces, we can really get structures of individual dimer interfaces, and then maybe are in a better position to explain why and how. But I think in the end, KDs and K-ons and K-offs of the back domain interactions and the BTB domain interactions will determine to a large extent the content of these granules, so how much P1 and P2 is going in, how much they like to form like homotypic clusters or heterotypic. So that's, yeah, it's a sort of information that we currently still just don't have.
Gotcha. Well, it's a great example of how sometimes structured domains drive droplets more than IDRs too. So
I think so. Yeah, I think that's, yeah, I was like when we, doing the first experiments, I was pretty simplistically thinking, we take out the IDR and the granules gone, but it was really the other way around.
but it turns out polymerization matters a lot too.
Yeah, definitely.
Any other questions? I guess not, but it's been a nice discussion with you, Rene.
Thank you.
Antonio, do you have anything else, or should I just close it down?
No, nothing else on this side except to thank Rene again, and great to see this work again.
Yes,
A pleasure.
100%.
Yeah.
Thank you so much for teaching us about the RNA and the granules and all the things. I know I learned a lot. So thank you everyone else for joining on Zoom and at the table and at the other table. Great to see all the faces, and we will be here in about a month again. So feel free to come back. Thanks again, Rene. Have a good one.
Thank you. Bye-bye.